I want to welcome everyone to Hawthorn University's webinar series. I'm Paula Bartholomew. It's my privilege to be the Director of Online Events and to welcome you and facilitate this presentation. Today we are going to welcome Tom Fabian to share his presentation on Microbial Mischief, the Microbiome-Autoimmune Connection. And I think this is such a timely topic since we have recent research that's implicated the microbiome as a potentially major contributing factor in the development of various autoimmune conditions with increased intestinal permeability and gut inflammation linked as well. So in this presentation, we're going to explore how these important factors are interrelated and how they can be assessed with the latest gut and microbiome testing methodology. And then with the information in hand, we'll discuss how microbiome-centered approach might be implemented to optimize nutritional support for individuals dealing with autoimmune conditions. I want to give you a little bit of background on Tom. He's a functional nutrition practitioner, an educator, and a speaker, as well as a research scientist with in-depth expertise in the role of the microbiome in health, disease, and aging. And he currently serves as a consultant for Diagnostic Solutions Laboratory. He has developed online educational programs for practitioners seeking in-depth education in the microbiome and related topics, and has extensive experience in the areas of aging and health span optimization. This is beautiful. I'm really honored to have Tom here and to be speaking on this really important topic. And we're going to get right into it here in a minute, but I also want to let you know that we'll have question and answer time at the end of the presentation, and we're going to go a full hour likely before we get to Q&A, so I wanted to let you know what your timing is, and we'll respond to your questions at the end, but please, by all means, write them into the webinar question panel at any time so that you're in queue and I'm prepared, and also that this presentation is going to be recorded and it'll be available for replay on our website in just a few days. With that in mind, hello, Tom.
Hi, Paula. Thanks so much for inviting me to speak today. It's great to be here.
It's really a pleasure to have you with us. I'm excited, and really, I'm just so eager to learn from you and your expertise that'll support us to understand this topic a little better, and most importantly, to really be able to actually implement this information. So thank you so much. Yeah.
My pleasure.
All right, I'm going to turn it over to you and let you take it and go.
All right, so as Paula mentioned, I'm going to be talking about the Microbiome-Autoimmune Connection today. So there's really a lot of new information that's been coming out about this connection, and I think there's a lot of implications, not only for helping to understand what's going on with patients and clients, especially though in terms of how to apply that in supporting our patients and clients. So that's really going to be the main focus of the presentation today. So first, before I dive in, I just want to talk about a couple of disclosures. So I'm currently a clinical education consultant for Diagnostic Solutions Laboratory. Some of you may be familiar with one of their main tests called the GI map. So that's one of the tests that we're actually going to cover a little bit about today. And also previously I was a consultant for two different companies, one of them Viome, which provides microbiome testing that is direct to consumer. And then Vibrant America, which provides a range of different types of testing supporting integrative and functional medicine practitioners. All right, so it's no surprise to anyone listening to this webinar that autoimmune diseases are on the rise, and they certainly are a big factor that is impacting health worldwide today. So just want to emphasize a few statistics to kind of underscore just how big the problem is. So the number of autoimmune diseases that are currently recognized is estimated to be in the range of 80 to 100 different autoimmune conditions. And then on top of that, there are a number of related conditions that are thought to have an autoimmune component. And so currently the estimate is that the number of those types of conditions is up to 40, but some of them are really major conditions, such as the various cardiovascular diseases. And in terms of numbers, the number affected in the US is estimated, depending on the data that's available and the definitions, again, because some of these are sort of in between, not necessarily well-recognized autoimmune conditions, but the numbers suggest that about 23 to upwards of 50 million people in the United States are affected by autoimmune conditions. And then in terms of the increasing prevalence globally, one study estimates that on average recently that the incidence of autoimmune diseases has been increasing by about 12.5% per year. So definitely a growing significant problem. So just want to kind of start off with a little bit of basics and definition of autoimmunity and what it involves, just to kind of orient everybody around this topic. So first of all, the immune system in autoimmunity attacks the body's own tissue. So that's obviously pretty well known. Tends to result in progressive damage to the target tissues. And then specifically, the pathology in most autoimmune conditions is thought to involve several things. First and foremost, it's thought to involve a genetic susceptibility. So there's definitely a genetic component involved. And then this is an area that's really relevant to what we can do therapeutically, is that there's a lot of environmental impacts on the development and risk for susceptibility for autoimmune conditions. And that includes obvious things like diet, lifestyle factors, et cetera. And then typically for a lot of autoimmune conditions, in conditions where this has been characterized, there sometimes can be a specific triggering event, such as a specific infection or toxin exposure. And then the pathology for autoimmune conditions also involves autoantibodies and what are called autoreactive immune cells. So basically, these are components of the immune system that basically are attacking our tissues. And then typically, kind of in a bigger picture, and this is something we're going to focus on quite a bit in this particular talk, because this is really where the microbiome imbalances come into play, is the balance between pro-inflammatory and anti-inflammatory factors. So that's thought to really set the stage for autoimmunity, as well as promote autoimmune conditions. And then basically all these together can create a vicious cycle. So with this imbalance, emphasizing pro-inflammatory type processes, that can ultimately result in tissue damage. And then both the tissue damage and the dysbiosis that's related to autoimmunity can promote further inflammation. So again, this creates kind of a vicious cycle, which is basically what's thought to contribute to that chronic progressive nature of autoimmune conditions. And this slide is basically just kind of a graphical summary of the various influences that are thought to lead to the development of autoimmune conditions. So kind of on the outside, we've talked about that at the bottom, you'll see the genetic influences, and at the top, you see the representation for environmental influences. So this is really setting the stage for the autoimmune conditions to be expressed in given individuals based on their genetics, based on their environmental influences. At their core, the various autoimmune conditions are thought to involve immune imbalances. So we talked a little bit about the pro-inflammatory, anti-inflammatory balance. That's really a key aspect to these immune imbalances that foster the development not only of autoimmune conditions, but also of chronic inflammatory conditions. And we also have the relationship of the immune imbalances to the microbiome and basically what's going on in the gut. So of course, that's going to be a big part of what we focus on today, is really this interrelationship between the immune imbalances and the microbiome imbalances. And so this is an outline of what I'm going to be covering in this webinar. And so based on what we just saw in terms of the key factors that are involved in autoimmunity, the first topic that we're going to cover is really diving into a little bit about what are those immune imbalances that are related to autoimmunity, and get into some detail there. And that's really kind of setting the stage for understanding how imbalances in the microbiome, which is known as dysbiosis, can contribute to autoimmunity as well. And then again, through that interrelationship between dysbiosis and the immune imbalances. And that's sort of the underlying science that we're going to focus on. So that will be a big part of what we're covering today. But I also want to get to the practical part. So that's really what the topics number 3 and 4 are all about. So in order to really understand what's going on with the microbiome, it can be very helpful to understand microbiome testing and incorporate microbiome testing in your practice. So we're just going to kind of touch on that topic and go through some examples. And then lastly, I want to wrap up with how does all this information come into play in terms of how we support and treat our clients and patients. So it really comes down to how do we promote a balanced microbiome. All right, so let's go ahead and start with this first topic. And we're going to focus again on immune imbalances related to autoimmunity. So it's important to note that there's essentially kind of 2 different categories of factors. So there's basically specific mechanisms that result in autoreactivity. And those mechanisms include things which some of you may have heard, for example, as molecular mimicry. We're going to talk just a little bit about that later on in the talk. And then really we're going to focus mostly on the general mechanisms, because that's really largely what's thought to come into play with regard to these microbiome imbalances. So these general mechanisms basically enhance any autoactivity that is present and result in chronic elevated inflammation. So this is something that can set the stage for autoimmunity, and then once autoimmunity develops, it can basically promote autoimmunity. So it's a very important aspect to understanding the whole autoimmune process, but also how do we deal with that. This is kind of a simplistic diagram, obviously, depicting just that balance between the pro-inflammatory aspect of the immune response and the anti-inflammatory aspect of the immune response. So again, this is a really finely tuned system. The immune system is very complex, and there's many things in many ways in which it can be disturbed. So it's really all about how do we get that perfect balance in the balance between the pro-inflammatory and the anti-inflammatory factors. And so we're going to dive into more of that balance and what that means. So essentially, when we're talking about pro-inflammatory responses, you can see on the left here, this involves two subtypes of immune response, by and large, that are referred to as Th1 and Th17 responses. This refers to a particular cell type that's basically an immune cell. So there's most of you have probably heard of T cells. These are T helper cells, and these are just different types of T helper cells. And so essentially they promote inflammation that ultimately can lead to autoimmunity. And essentially in various autoimmune conditions, these types of responses, Th1 and or Th17, tend to be both elevated in a variety of autoimmune conditions. And then on the flip side of that, we have the anti-inflammatory responses, known as Th2, and also a fairly more recently recognized category called T-regulatory responses. And these tend to be quite important for helping to restrain inflammation and helping to reduce the incidence of autoimmunity. And essentially they promote immune tolerance. And we will talk a bit more about that as we proceed as well. So essentially I really want to focus mostly though on the Th17 Treg balance, because it's thought that that is really what comes into play in mucosal tissues, especially in the gut. So that's really the most relevant aspects of this balance when it comes to the gut microbiome. So just a little bit more information about this Th17 T regulatory balance. So this is a review article titled "The Balance of Th17 versus Treg Cells in Autoimmunity." And in their abstract, they say, accumulated evidence suggests that the Th17 Treg balance underlies the pathogenic mechanisms driving autoimmune diseases. So this is, again, really coming out of recent research in the past 5 to 10 years or so, emphasizing that this appears to be a very important component of autoimmune conditions. They go on to say therapeutic approaches designed to correct the balance have proven effective and have been approved for the treatment of rheumatoid arthritis, psoriasis and psoriatic arthritis, ankylosing spondylitis, systemic lupus erythematosus, and other autoimmune conditions like MS and IBD. So essentially, not only has this been recognized as being a factor in a number of these different conditions, but new therapies that have been based on this imbalance have been shown to be effective. So we know that they are definitely involved and that we can use that information to help improve symptoms and the lives of patients. So basically, this can kind of be summarized in the concept of the yin-yang relationship. So Th17 cells essentially represent one side of that relationship, whereas T regulatory cells represent the other side. And again, the idea is that these two need to be in balance. We actually do need some level of the pro-inflammatory response so that we can respond appropriately to various infections, pathogens, etc. We need to be able to deal with those sorts of threats and insults, but we also need to restrain those responses so they don't become out of control. So again, this is really all about achieving that balance, and how do we do that? In this article, which is titled "When Worlds Collide, Th17 and Treg Cells in Cancer and Autoimmunity," gets into a little bit more detail here. So they say, while the division of helper T cells, those are those Th cells, while the division of helper T cell support was previously hypothesized to be dominated solely by Th1 and Th2 helper subsets. This is just a side note that many of you have probably heard of Th1, Th2, and especially the Th1, Th2 balance. Again, this is where the new research has come into play, where we're learning more about the Th17 and Treg balance as being part of that equation as well. So they go on to say, mounting evidence over the past decade reveals that Th17 cells and regulatory T cells, or Tregs, also play important roles in regulating health and exacerbating autoimmunity and cancer. This is a table from that article, and it's obviously kind of a complicated table. So you'll see the area in red. I'm just going to focus in on this slide. So essentially, if you look at the top of the two columns, the first column is Th17's role in pathogenesis, and then the second column to the right is the Treg role in pathogenesis. And then the column to the left lists various autoimmune conditions where these types of responses have been characterized. So you'll see that basically unanimously in that Th17 column that all these conditions have been associated with increases in Th17 type responses. And in particular, Th17 cells produce a particular cytokine called IL-17, which seems to play a very important role in chronic inflammation as well as autoimmune pathogenesis. And then on the right, you'll see the results for Tregs in these various conditions, and in most cases, Tregs are reduced. So you can imagine when one is increased, the other one is decreased, that really sets up a situation where you have a significant imbalance and allows essentially kind of a runaway inflammatory process. Let's talk a little bit more about the Th17 responses. So basically the majority of Th17 cells reside in the gut itself. In particular, most of them reside in what's called the lamina propria. Their function is to protect against extracellular pathogens by promoting inflammatory responses. So that's their normal function, usually non-pathogenic. So essentially, they counteract pro-inflammatory bacteria, for example, such as Klebsiella or Salmonella and many others. And they also tend to counteract, in particular, Candida. So it can be very important for containing and reducing Candida infections. But an excessive Th17 response contributes to autoimmunity. So that should be pretty clear at this point based on the information we've covered. In terms of Treg responses, the majority of Treg cells also reside in the gut, again, in particular in the lamina propria. They tend to suppress inflammatory responses, including those mediated by Th17 cells. And then they also promote immune tolerance in several ways. They promote immune tolerance to self-antigens, which of course is the most relevant for autoimmune conditions, but they also promote tolerance, essentially a lack of immune reactivity, to the beneficial commensal microbes. Obviously, we don't want to get rid of those beneficial commensal microbes. We want to restrain the immune response. And then Treg cells also promote immune tolerance against food antigens and other environmental antigens. So they really have an important role in helping to keep our immune system from being overreactive to things that it doesn't need to be reacting to. Okay, so back to our diagram of the balance between pro-inflammatory and anti-inflammatory responses. So I want to focus in on the microbiome imbalances. So part of that equation. So we just talked about the immune imbalances and kind of got a sense there, especially of the importance of Th17, Treg cells. Now I want to talk about the connection to the microbiome imbalances. So that's this next section in the presentation. So just to kind of orient us around the importance of the gut. So of course, many of us in nutrition and functional integrative medicine are well aware of the importance of the gut for health. But in particular, in relation to between the microbiome and the immune system, it's important to understand that especially the lower GI tract is home to over 100 trillion microbes. So it's a very large number of microbes that are present that essentially the immune system has to figure out how to deal with and differentiate, especially beneficial from potentially harmful microbes. And that's essentially why we have 70 to 80% of the immune system in the gut. Because there's basically largely this huge presence of microbes that are also present in the gut. And on top of that, of course, we know that there's a very large epithelial surface area to the gut. And that's partly to facilitate efficient absorption, but that creates an issue where we have this really large surface area that essentially the immune system has to monitor. So basically, it's involved in monitoring for any sort of pathogens that may be present, but also tending to the beneficial bacteria that are there. This is zeroing in on that intestinal mucosal layer. This is basically just to give you a little bit of a visual when we zoom in to really emphasize how in close proximity the microbes are to the immune system. So you can see here in the diagram that kind of in the top part is representing the lumen. Actually, let me go back. So the top part is representing the lumen, and then you'll see that they're the intestinal microbiota that are very closely associated with the single layer thick epithelium. So that's really only one cell layer that's separating the microbes from the underlying immune system. And so the immune system and the epithelium work very hard to keep those microbes contained where they are, even the beneficial ones, because we know that once they interact with the epithelium, once they interact with the immune system, that can set off a response. You'll note in between that there is a protective mucus layer. Part of the role of the mucus layer is to help separate the bacteria and the other microbes, pathogens, and commensals from interacting with the epithelium as well as the immune system. And again, that's one mechanism to help to keep the immune system from responding excessively. But there's really a huge presence of immune cells and immune factors, basically in close association with the epithelium, and they're constantly monitoring what's going on in the lumen with regard to microbes and pathogens. All right, so I just want to talk a little bit about the relationship between microbes and the immune system. And this article here summarizes some of the key concepts. The article is titled Impacts of Microbiome Metabolites on Immune Regulation and Autoimmunity. I just want to emphasize a little excerpt from the abstract. So they say that specific components of the gut microbiome have been implicated in the production of pro-inflammatory cytokines and subsequent generation of Th17 cells. So essentially they're emphasizing that these various potentially inflammatory products produced by the microbiome, including things called LPS, which stands for lipopolysaccharide, tend to be very pro-inflammatory. They go on to say, similarly, commensal bacteria and their metabolites can also promote the generation of regulatory T cells, Treg cells, contributing to immune suppression. So that's really summarizing that direct link between the microbiome, what they produce, and the influence of those products on the balance in the immune system. And we will be going into some of those details here shortly. So back to our slide summarizing the influences that lead to the development of autoimmune conditions. And I really want to emphasize this two-way arrow in between the microbiome imbalances and the immune imbalances. I want to give a couple examples of how each factor, the microbiome, can influence the immune system, and the immune system in turn can influence the microbiome. And that's an important concept to understand when we're learning how to work with managing the microbiome. So this article is titled Microbiota, an Amplifier of Autoimmunity. And they say in the article, immunoglobulin A, IgA, is one critical adaptive immune effector molecule that maintains a diverse and balanced bacterial community in mice and humans. So it's needed for normal balance in the microbiome. IgA coating of indigenous bacteria promotes stable colonization of symbionts while driving immune exclusion of enteric pathogens. So immunoglobulin A is really one key factor produced by the immune system that helps basically tend to the beneficial bacteria and then reduce the incidence of pathogens. So it's one key factor that helps to balance the microbiome. And they go on to say patients lacking IgA suffer from an increased incidence of immune diseases, suggesting the fine-tuning of the microbial community by IgA prevents inflammation or loss of tolerance. So this is an important aspect potentially to understand in terms of autoimmune conditions. All right, so back to our diagram that's basically emphasizing the balance that's needed between the pro-inflammatory responses and the anti-inflammatory responses. I just want to emphasize that the pro-inflammatory responses tend to be promoted by inflammatory microbes, and then the beneficial microbes tend to promote anti-inflammatory responses. This is really kind of the link that we want to focus on. So now I want to go through examples of both the inflammatory and beneficial microbes and give you a sense of what they're doing that could potentially influence the immune system. So in terms of the inflammatory microbes, let's just talk about some of the key impacts that they can have. So they can definitely influence the balance of Treg and T17 cells. And in particular, the inflammatory microbes can reduce beneficial microbes. So basically they kind of have this seesaw relationship. When the inflammatory microbes tend to increase for whatever reason, then that causes a decrease in beneficial microbes. So right there, we're getting an imbalance in the microbiome that can then lead to this immune imbalance. They also tend to promote inflammation autoimmunity, especially through the Th17 type responses. They also tend to promote a healthy intestinal barrier. At least the beneficial ones do, but we're talking about the inflammatory ones. So essentially they promote an increase in intestinal permeability and also barrier dysfunction. So that's really a key aspect to note about inflammatory bacteria, is that not only can they cause these immune imbalances, but they can also contribute to leaky gut and other types of barrier dysfunction. And then some of them actually may cross the intestinal barrier, and they can enter circulation. Sometimes they can invade tissues, and then they can cause either localized infections, or they can just promote a localized immune response or result in inflammation. All right, so next I want to talk about, the previous slide was really emphasizing some of these broader aspects in how the microbiome can influence the immune system, again, on a more broad basis. But we also know that some of these microbes actually can influence autoimmunity by specific mechanisms. So this is basically just talking a little bit about some of these autoimmune specific mechanisms. So we know that molecular mimicry is involved in some cases. And for those of you who are not aware of what that term means, basically it involves the fact that some microbes can produce proteins that have a similar structure to structural aspects of human proteins. So it doesn't necessarily mean the whole protein is similar, but there can be parts of it that are recognized by antibodies that are similar. And so basically they promote antibody-mediated inflammatory responses that can cross-react with those human proteins. So they basically cause an immune response against these human proteins. And then other types of bacteria can actually produce enzymes and other factors that can modify human proteins. So essentially what happens is these modified proteins then are seen as foreign by the immune system. So they promote antibody-mediated inflammatory responses against those proteins. There are other proposed mechanisms for how various microbes may promote autoimmunity on a very specific level. These are just two of the more well-characterized type mechanisms. So I just wanted to highlight a few examples of these autoimmune-associated microbes. So we're going to be talking a little about Klebsiella pneumoniae, but that's one that's been linked to a number of different, not only chronic inflammatory conditions, but also several autoimmune conditions. Prevotella copri, which has been associated with rheumatoid arthritis. Streptococcus species, which have been associated with certain immune conditions and autoimmune conditions as well. Citrobacter, it's actually misspelled, should be C-I-T-R-O, Citrobacter. And then Helicobacter pylori, which is fairly common, has also been linked to various autoimmune conditions, including in particular autoimmune thyroiditis. And then an oral pathogen that tends to be involved in periodontitis is Porphyromonas gingivalis, and that has also been linked to various autoimmune conditions as well. All right, so now I want to zero in a little bit and talk more about Klebsiella as an example of an organism that actually can contribute to both the general imbalances related to autoimmunity as well as some of the specific imbalances. So it's been linked to several autoimmune conditions and inflammatory conditions as well, as I mentioned. Typically resides in the oral cavity as well as the respiratory tract. So it's not always a resident of the gut. It can be in a lot of people, but it's more common to be found in the oral cavity, respiratory tract. And so it's thought to colonize the gut when certain protective factors are compromised, especially when there's hyperchlorhydria or low stomach acid, as well as existing gut dysbiosis. And especially when beneficial bacteria are relatively low and deficient, that can allow the niches to open up for Klebsiella to colonize. Tends to promote an inflammatory response in the gut that's pretty well characterized. And then it also may promote autoimmunity via molecular mimicry mechanism. And so that's emphasized here in this particular article. So the title is Antibodies Directed Against a Peptide Epitope of a Klebsiella pneumonia-derived protein are present in ankylosing spondylitis. So I just want to highlight a couple of things from this abstract. So several reports have implicated Klebsiella pneumoniae as a triggering or perpetuating factor in ankylosing spondylitis. They go on to say that serum IgG antibodies against a particular Klebsiella protein component were detected in 190 out of 200 patients. So basically 95% had these antibodies against Klebsiella. Without going into too much detail, they did basically link those antibodies to a particular protein in humans that's basically very similar. It has a very similar structure to this protein in Klebsiella. So it's thought that there's a cross reaction then of this anti-Klebsiella antibody to the human tissues. And then they go on to say that such reactivity was not detected in healthy donors. So basically 95% of people with ankylosing spondylitis had these antibodies, and no healthy donors actually had these antibodies. So it seems to be a significant differentiating factor. Our results show that antibodies directed against an epitope of the Klebsiella pneumoniae-derived protein are present in nearly all patients with AS. So there's definitely a link there between Klebsiella and the potential for molecular mimicry involved in ankylosing spondylitis. And then in this second article, the title is Rheumatoid Arthritis. Rheumatoid arthritis patients exhibited impaired Candida albicans specific Th17 responses. So they say accumulating data implicate what are called the CD4 T cell subsets, which is basically the scientific name for Th17 cells in rheumatoid arthritis, and that the Th17 IL-17 axis is vital for immunity to fungi, especially the commensal fungus Candida albicans. So we talked about this Th17 pro-inflammatory response isn't necessarily a problem, except when it's out of balance. And so normally it's needed to basically help keep these various opportunists and pathogens in check. So again, it seems to be very important for protection against Candida albicans infection. They go on to say that significantly more rheumatoid arthritis patients were colonized with Candida albicans in the oral cavity than healthy subjects. So there's basically, they're characterizing an association here between rheumatoid arthritis, Th17, and the presence of Candida albicans. This is just basically a figure from another article that talks about this connection between Th17, IL-17, and protection against Candida a bit more. So just wanted to basically show this so we have kind of a visual representation of that. So you can see at the top, basically that the blue cells represent the epithelial layer, and then the area above that is the intestinal lumen, and then the purple cell-looking things with the projections, that's representing Candida. It's basically transformed into the form that creates these hyphae that can penetrate the epithelial layer. That's really what gets inflammation going during a Candida infection or overgrowth. Again, when these hyphae tend to penetrate the epithelial layer, they can basically promote inflammation, and they can also promote leaky gut. And that's largely carried out through kind of this complex sequence of events that essentially lead to the production of these Th17 cells, that's in the highlighted area, circled in red, that are basically producing various factors, including the cytokine IL-17, that basically then promotes the activity of inflammatory cells such as neutrophils that normally under normal conditions help to reduce Candida. They basically can attack the Candida and then control the inflammation or the infection. But if they're out of control and things are imbalanced and you have an excessive Th17 response, that can produce an excess of neutrophils that can result in tissue damage. All right, so back to our diagram illustrating the balance between inflammatory microbes, beneficial microbes, and their impact on the immune system. So now we're going to focus on the beneficial microbes. So let's talk a little bit about the beneficial microbes and their key products. So they tend to keep pathogens and opportunists in check. So that's one of their most important functions in the gut microbiome. So when they're deficient, as you can imagine, that's going to lead to a situation where we don't have sufficient protection against pathogens and opportunists. They also tend to promote a healthy intestinal barrier and reduce leaky gut. That's one of their really very important functions. So when we're thinking of how do we address leaky gut, instead of always thinking specifically in terms of what that means in terms of supplements such as L-glutamine, we should also be thinking about what other things may be going on. Are there beneficial bacteria deficient, and which ones are deficient, and then how can we support them? And then they're also involved pretty critically in helping to reduce inflammation and promote immune tolerance, again through the promotion of T regulatory cells. So if you're interested in learning more about how beneficial bacteria basically can support the intestinal barrier and help to reduce inflammation, this is a good recent review article. And as you'll note at the top, there's an icon that says PMC, full text free. So that's basically an indicator that you can read the entire article for free. So I would suggest that if you're interested, again, in this connection between pro-inflammatory, anti-inflammatory bacteria, this is a great article to check out. But also I want to emphasize this just because the title itself, the potential of gut commensals in reinforcing the intestinal barrier and alleviating inflammation. Those are really two key aspects to potentially addressing a wide range of inflammatory conditions and autoimmunity. So really it can't be overemphasized just how important these beneficial bacteria are. Let's go through a few examples here of important beneficial bacteria. So a lot of people would include butyrate producing bacteria at the top of the list. There's lots of research around these bacteria and their beneficial effects. They tend to be in a group called Clostridia, and there are a number of different types of species that can produce butyrate. And basically, here's just a short list of some of the important ones. So there's something called Faecalibacterium, Roseburia, and also Eubacterium, and then many others as well. And then, of course, when we think of beneficial bacteria, we tend to think of probiotic bacteria. And that typically includes species like Lactobacillus species, Bifidobacteria species. Just as a side note, you'll see this SPP abbreviation quite a bit. That just stands for species. And that's basically what's used in scientific notation to indicate multiple species. And there's also the Bacteroidetes group, which is essentially a whole phylum of bacteria. And there's a number of different Bacteroidetes that are known to be important and have been characterized in terms of helping to promote a healthy intestinal barrier, as well as helping to reduce inflammation. Especially Bacteroides fragilis and Parabacteroides distosanus have both been characterized in terms of helping to promote those Treg cells. All right, so let's go a little bit more detail into the example of butyrate. So it's produced primarily by certain Clostridia species, as I mentioned. It reduces intestinal permeability and promotes intestinal barrier health. In particular, it can help promote the production of that protective mucus layer, as well as secretory IgA. So we talked about how important those are. Also promotes production of Tregs, and they also serve as a primary source of energy for colon cells, which turns out to, through kind of a complicated mechanism, which I won't get into, promotes an anaerobic environment for beneficial bacteria. So just kind of a quick side note is that by and large, the beneficial bacteria in the colon are anaerobic. They prefer an anaerobic environment. They don't do well when there's certain levels of oxygen present. On the other hand, the inflammatory bacteria tend to do better in an environment that has a little bit more oxygen. So essentially, this is really helping to keep an environment, set of environmental conditions that are more conducive to the beneficial bacteria and not so conducive to the pro-inflammatory bacteria. Just a couple of studies to kind of emphasize the importance of butyrate for especially immune balance. So this title is for this article is Gum Arabic Fibers, which is a type of prebiotic, decreased inflammatory markers and disease severity score among rheumatoid arthritis patients in a phase 2 trial. So just going to quickly go through this here. Not going to really go through the whole study. Just want to emphasize a couple things from this. Gum arabic fermentation by colonic bacteria increases serum butyrate concentrations, so it's considered as a prebiotic agent. Gum arabic has anti-inflammatory activity through its derivative butyrate. And then this is a clinical phase 2 trial with 40 patients. And basically what they found is in this study, they showed this prebiotic fiber that promotes butyrate production resulted in a decrease in the level of serum TNF-alpha, which is a marker for inflammation. So it reduced inflammation. And then it also reduced the number of swollen and tender joints in rheumatoid arthritis patients after 12 weeks of taking this particular prebiotic. So there was significant alleviation of symptoms in this study. Another one, this basically, the study is titled Attenuation of Rheumatoid Inflammation by Sodium Butyrate through Reciprocal Targeting of HDA2, HDAC2, and osteoclasts in HDAC8 and T cells. So that's a lot of kind of technical jargon, but I'll just get to the point here that they evaluated therapeutic effects of butyrate. And again, this is butyrate supplied as a supplement, not by applying prebiotic. So even supplemental butyrate has been used and can be beneficial. But basically, they say they evaluated therapeutic effects of butyrate in an animal model of autoimmune arthritis. The arthritis score and incidence were lower in the butyrate-treated group compared to the control group. So this had a positive benefit in an animal model of autoimmune conditions. They go on to say these findings suggest that butyrate is a candidate agent for the treatment of RAA. So again, butyrate, apparently in several different forms. It can be generated locally from prebiotic fibers, but also can be supplemented and have a positive effect as well. So I want to go through one more example of the relationship between certain beneficial bacteria and autoimmunity. This example involves basically dietary sodium and how high sodium intake can promote autoimmunity by reducing beneficial bacteria. So this review article is titled "The Role of Dietary Sodium in Autoimmune Diseases, the Salty Truth." Basically summarizes this relationship. So they talk about high salt intake may play a role in the development of autoimmune diseases. Several lines of evidence point toward the role of high sodium intake in reversing the suppressive effects of regulatory T cells or Tregs, and instead promote cellular shift towards T helper 1 and Th17 pro-inflammatory phenotypes. So salt in the diet can actually promote a more inflammatory type balance in the immune system. And then this article basically talks about the involvement of lactobacillus. So the title is Salt Responsive Gut Commensal Modulates Th17 Axis and Disease. And in this abstract, they say, here we show that high salt intake affects the gut microbiome in mice, particularly by depleting Lactobacillus murinus, which is a specific species of lactobacillus present in mice. Consequently, the treatment of mice with L. murinus prevented salt-induced aggravation of actively induced experimental autoimmune encephalomyelitis, which is a model for multiple sclerosis, and then salt-sensitive hypertension by modulating Th17 cells. And they go on to say, in line with these findings, a moderate high salt challenge in a pilot study in humans reduced intestinal survival of lactobacillus species, increased Th17 cells, and then increased blood pressure. So they're showing that basically this also seems to apply in humans and results in a reduction of certain sensitive lactobacillus species. So lactobacillus turns out to be quite important also in relation to protecting against autoimmune conditions. Again, so we're kind of back to illustrating our balance here. We talked about the role of pro-inflammatory microbes and the role of beneficial microbes. So just want to kind of mention as a side note, this is a common sort of model that I use in a lot of my courses and lectures, illustrating the interrelationship of the microbiome with immune function and the intestinal barriers. So we went through a lot of examples here, so you should really start to see that there's this extensive interconnection between these components. So that's important for understanding disease pathology, but it's also very important for understanding how we might approach autoimmune diseases in terms of therapeutic effects. All right, so just want to kind of quickly go through the microbiome testing section so we can spend more time on the implications for therapy. Just so you're familiar with some of the options that are out there and sort of what they may be beneficial for. So there are quite a few options now for microbiome testing. There's really two different types. There's direct-to-consumer testing. Those types of tests give you kind of an idea of the general microbiome composition or function, and they also tend to be wellness-related interpretations. So they're not necessarily geared towards clinical applications. They're really geared more towards information that may help in terms of understanding dietary impacts, et cetera. Then there's the more clinical testing that's available. That really focuses more on pathogenic organisms as well as opportunistic organisms. Does often give us some sense of the general microbiome, but it was really focused on these more disease-related microbes, as well as providing information related to gastrointestinal health through various markers. So again, that's really where it becomes much more clinically applicable as opposed to the direct-to-consumer testing. So this is an example of one of the direct-to-consumer tests out there that's pretty common. This is essentially a screenshot from uBiome, and these are my recent results from uBiome. So you can see basically when you log in, you see kind of a dashboard page that gives you your wellness score, your diversity score, and then there's various sort of health-related categories where you can look to see how your microbiome compares to sort of the average population, whether you're healthier, less healthier, in relation to these various sort of wellness-related markers. So that some of that information can be useful. It's not necessarily always sort of the most clinically relevant information, but certainly can give us some insight into how our microbiome relates to our particular health issues. It's just an example of one of those categories, healthy weight. So you can see how I compare to the average. So I'm slightly less healthy in terms of my microbiome compared to the average according to this particular comparison. And then you can dive in into much more depth for those of you that want to kind of get into these details. So there is a section where you can actually see the various taxonomic levels of microbes that are present in your microbiome. And of course, the main caveat here is to begin to understand the implications of this information. You really have to learn a lot more about the microbiome and what the various functions are of these various groups, so you can start to understand the impact of what the composition may mean for health. And you can also download your data. So if you really want to get into the detail, you're able to actually get data down to the species level. There's a lot of information here, again, so that really takes kind of a certain level of expertise to really work with that information. So again, a lot of people that do use the direct-to-consumer testing tend to stick with the sort of information that's already interpreted for you and related to wellness. This is an example of a clinical test. This is from a company, Diagnostic Solutions. So I mentioned at the beginning that I consult with Diagnostic Solutions. Their test is called GI-MAP. This is basically a stool test that gives you an idea of first off pathogens. You can get an idea of what pathogens may be present. And then you can see other opportunistic organisms like parasitic pathogens, viral pathogens, H. pylori. You can see a little bit about your normal bacterial flora, especially focusing in on the phylomicrobiota, where we can get a lot of information on the beneficial bacteria. There's a section for opportunistic bacteria, including a section on potential autoimmune triggers. So this involves some of those organisms that we talked about that have been specifically associated with certain autoimmune conditions. We just talked about Klebsiella pneumoniae. I just wanted to emphasize that this test does include Klebsiella. Candida we also talked about. That's also included on this test, as well as additional viruses that have also sometimes been linked to certain autoimmune conditions. And then we have a range of intestinal health markers. So it's just kind of quickly going through this test here to let you know that there's these various types of markers that we can get information on. I'll actually go through an example later on in terms of how we can potentially use this information to inform our therapeutic approach. So just quickly summarizing the challenges of microbiome testing. Basically, there is a moderate to steep learning curve to learn how to interpret and connect the dots. That really can't be emphasized too much if you really want to dive into microbiome testing, especially the direct-to-consumer testing, and you want to get into those details. It does take a certain level of expertise to understand that. But basically, there are an increasing availability of learning resources out there to help you learn about those various microbes and how to interpret them. But also, you have the option of partnering with a knowledgeable practitioner or a subject matter expert to basically help you with that interpretation of those tests. There's also some controversies in terms of the underlying technology. Different companies use different approaches, and there is debate over which technologies are better than others, as well as how to interpret that information. There's some debate over that as well. It can involve a significant time investment to analyze the results for each individual patient. So that is something to take into account. And then, of course, the test itself has a cost to it, so there's additional costs for clients and patients. But also just want to emphasize the other side of the equation, the benefits of microbiome testing that can help identify general microbiome imbalances and possible specific contributors to symptoms. So that's a really important aspect to microbiome testing because we know gut testing is so important, or the gut itself is so important for understanding the health of our patients and clients. Clinical testing also provides insights into gut and immune function, especially digestion, inflammation, leaky gut, etc. It often provides really important insights excuse me, into an individual's underlying imbalances and can really help us connect the dots. So that's something I really want to emphasize, again, because we know that the digestive tract is so important, especially when it comes to autoimmune conditions. We know that there's this major connection between the gut and the immune system. And so gut testing really can give us some actionable information. So that brings me to our last section on how do we use this information to promote a balanced microbiome. So just a few sort of general notes about promoting a balanced microbiome. Generally, this sort of involves several pillars that we want to make sure that we're emphasizing. Basically, it took a fair amount of time in this webinar to emphasize the importance of the beneficial microbes. So it is important to support them. It's also potentially important to inhibit or eliminate those potentially harmful microbes, the pro-inflammatory ones. We also want to promote a healthy intestinal barrier and also promote optimal digestion. Just kind of a side note, I'll talk a little bit more about this later, but it turns out that digestion is also a significant driver of dysbiosis. So we want to make sure that digestion is optimal, particularly if we're not digesting proteins and carbohydrates as well, that can feed imbalances in the microbiome. So this is just sort of emphasizing that most practitioners, when they think of how do you promote beneficial bacteria, first thing they tend to think of is probiotics. So I'll talk a little bit about that. So many studies, of course, do support their beneficial effects, but individuals really vary a lot in their response. That's something we see a lot clinically, and there's also research showing that there can be really vast variability among individuals and how they respond to probiotics. So it is something to take into account, and typically you have to do a bit of trial and error with probiotics to see what may work for a given patient or client. Obviously, most of us know the sort of common examples of probiotic bacteria, such as Lactobacillus, Bifidobacterium, the Bacillus species, which are generally known more as spore-forming or soil-based species, Saccharomyces boulardii, and also kind of an up-and-coming area for probiotics are the bacteriophages. So there's a growing number of different products that are available now that offer those. Basically, overall, though, when we're considering probiotics, we want to consider that they're promoting the function and colonization. Actually, we want to promote their function and colonization. So it's not just sufficient to supply probiotics, but we also want to create those conditions that allow them to more likely flourish in the gut and to do the beneficial things that they need to do. So it's been shown in many studies that supporting them with prebiotics can be helpful. There's new information suggesting that polyphenols can also be helpful to promote their function. A low-salt diet, as we saw, can be helpful particularly to promote lactobacillus. And then tryptophan, making sure that there's enough essentially protein in the diet, especially tryptophan, because some of those lactobacillus species can actually convert tryptophan into beneficial products. And of course, general gut support, because we know that the probiotic bacteria depend heavily on a healthy intestinal lining. So for promoting beneficial commensals, we want to focus on prebiotics, basically to bring up the butyrate producers. In particular, there's good evidence that inulin-resistant starch and even the supplement butyrate itself can in turn promote the butyrate producers. Then in terms of promoting the Bacteroidetes, which is another major beneficial group of bacteria, a prebiotic called glucomannan can be helpful, as well as intermittent fasting, which has been shown to increase them. Phytochemicals and polyphenols can be very important, especially curcumin, quercetin, polyphenols from berries, and many other sources like pomegranate, green tea, and cocoa, and of course intestinal barrier support. I just want to talk a little bit more about phytochemicals and polyphenols and how they're really important potentially for balancing the microbiome. So this is a review article that was published recently, and in the article, I just want to emphasize this one line where they basically conclude from looking at all these various studies on polyphenols in the microbiome that they tend to inhibit pathogenic bacteria and they stimulate beneficial bacteria. So again, they can be very helpful towards helping to balance the microbiome. And this is a specific example of what's known about curcumin, summarizing its effects in relation to counteracting inflammation. So it's just basically a table from this particular article called Curcumin as Curcumin from Kitchen to Clinic. And essentially it's showing all the different known pro-inflammatory factors that are inhibited by curcumin. So again, curcumin is a very well characterized anti-inflammatory. So inhibiting harmful microbes, antimicrobial botanicals can be very important. Promoting the beneficial commensals is something that's often overlooked. We tend to go right to the antimicrobials and sort of forget that really one of the main roles of the beneficial commensals is to protect against the harmful ones. And that includes probiotics. Just kind of in the interest of time, kind of want to skip over this part a little bit. Immunoglobulins can be quite important. There are various sources now. So colostrum is a great source, but for those who are Sensitive to dairy, there are serum bovine-based products that are dairy-free. Saccharomyces boulardii and bacteriophages can be helpful in certain cases to inhibit harmful microbes, as well as anti-biofilm agents, since we know that some of these harmful microbes produce biofilms. And then also, as I mentioned, addressing digestive dysfunction, which tends to feed the harmful bacteria. So promoting a healthy intestinal barrier, basically that involves addressing beneficial and harmful microbes, since they basically the harmful microbes, of course, can disrupt the barrier. Beneficial microbes can promote a healthy intestinal barrier. So really that's a key focus for restoring a healthy intestinal barrier. But even the short chain fatty acids that they tend to produce themselves can be important, such as supplementing with butyrate or propionate can basically help promote a healthy intestinal barrier. Immunoglobulins, once again, have been shown to be very beneficial in this circumstance, along with certain supplements like L-glutamine, zinc carnosine, and acetylglucosamine. Vitamins A and D are really important for the healthy intestinal barrier and also addressing inflammation with omega-3s. I'll skip over this because it's kind of a detailed thing, but certain other supplements can be quite helpful to promote the barrier. And then I can't be emphasized enough really in terms of not only focusing on optimizing diet and optimizing the supplement protocol, but really focusing on optimizing digestion because I can't overemphasize just how important that is for microbiome balance. Just want to mention here real quick, I won't go through these, but basically several studies have shown that various types of fasting actually may be quite helpful in inflammatory conditions and possibly for autoimmunity as well. The title here is Intermittent Fasting Confers Protection in CNS Autoimmunity by Altering the Gut Microbiota. I want to skip that and just kind of emphasize that there's some examples here. Another type of fasting protocol called the fasting mimicking diet is known to promote regeneration and reduce autoimmunity and multiple sclerosis symptoms. And this was done mostly in an animal model. So lots of promising research around fasting as another therapeutic option. So I just want to quickly end with going through an example here of a particular set of test results and how we might use those results to promote gut health. All right, so in this example, we're going to be looking at microbiome clinical test results, not a direct-to-consumer test, and also looking at how we would address those imbalances with diet and supplements. So basically, this is sort of illustrating kind of a common set of circumstances that we tend to see in gut testing in terms of the common imbalances. So oftentimes we'll see H. pylori is positive, which you can see here in the GI map test from Diagnostic Solutions Lab. We do know that H. pylori potentially can cause upper GI issues, sometimes serious. That's what the virulence factors are listed here to help clinicians better understand the pathogenic potential. But even H. pylori by itself, when it's high, does have some impacts. One of the best known impacts is in potentially reducing stomach acid or causing hypochlorhydria. Then in the normal bacterial flora section in this example, we'll see that under the phylomicrobiota, both the Bacteroidetes and Firmicutes are low. And again, those are two main groups of bacteria. Actually, let me back up here. So there are two main groups of bacteria, and together they make up roughly about 90% of the microbiome. So again, that's the Bacteroidetes and Firmicutes. So when both of those are low, that's telling us that overall the microbiome is deficient in beneficial bacteria. And when we look at the opportunistic bacteria section, we can see that several different organisms are overgrown. Basically, and it's sort of beyond the scope of this webinar to get into the details here, but this particular pattern basically reflects what we would consider a lack of stomach acid and reduced digestion. So we can connect that to the presence of H. pylori. This is something that we often see as a pattern with GI map, basically where these particular types of organisms are overgrown and they are indicative. That's not only something we see clinically, but also something that's been shown in research that many of these tend to be elevated when stomach acid is low and digestion is reduced. You'll also note here in these results that Klebsiella pneumoniae, which we talked about, is elevated, and that's highlighted here. So if this were an autoimmune condition, that would be something we want to focus on as a potential, playing a potential role depending on what the condition is. So in this case, we also often see Candida overgrown when digestion is not optimal. That makes a lot of sense when we think of, what are the circumstances that cause Candida to proliferate. Typically, those are things like a high-carb diet, or when carbohydrates at sort of whatever level they're consumed are not well digested. Those scenarios often tend to overfeed Candida and fuel the overgrowth. So we often see Candida elevated when H. pylori is present. This is an example of the intestinal health markers that we often see in this scenario. So you'll see, based on what we already looked at before, that we know when there's low levels of beneficial bacteria, that's going to have an impact on the intestinal barrier and result in a sort of poorly functioning intestinal barrier. So that can lead to increased reactivity to antigens. So that includes foods, that includes microbes. So secretory IgA can reflect that. So when we see elevated secretory IgA, that often goes along with a poorly functioning intestinal barrier that's letting stuff get in contact with the immune system and causing a response. In this case, there's also elevated activity anti-gliadin IgA, which is basically a marker for reactivity to gluten. So in this case, of course, we want to follow up and see if the patient or client is consuming gluten, and if they're not, there may be cross-reactive foods. And then with regard to inflammation, calprotectin is a great marker for intestinal inflammation. And of course, in this case, it's showing that the level of inflammation is elevated. And that's something we commonly see when there's pro-inflammatory dysbiosis and or the presence of Candida. And it is known that Candida can be pretty inflammatory. So the conclusions here is that we have H. pylori, and that's probably contributing to hyperchlorhydria and downstream dysbiosis. We have low beneficial bacteria, especially the Bacteroidetes and Firmicutes, so that implies intestinal barrier dysfunction, lower anti-inflammatory capacity, as well as the reduced ability to inhibit the pathogens. So again, we really want to work on those beneficial bacteria, high opportunistic dysbiotic bacteria, and Candida. So basically that's consistent with a digestive dysfunction pattern, typically involving low stomach acid, a pro-inflammatory pattern, as well as consistent with intestinal barrier dysfunction. So this is fairly typical to see this kind of extensive dysbiosis, especially when certain kind of upstream pathogens are present like H. pylori or possibly Giardia. In addition, we saw that in this case, actually, elastase was normal. That's a marker for digestion. So it looks like pancreatic output is normal in this case, even though stomach acid may be low. We saw high secretory IgA, high anti-gliadin IgA. So again, that suggests that there are opportunists present. There's likely some food reactions, reactivity to gluten, poor barrier function. That's also often linked to Candida when we see high secretory IgA, and also implies that there may be SIBO. So that's something to consider, obviously, when there are symptoms related to SIBO. Elevated calprotectin suggests gut inflammation, possibly due to pro-inflammatory dysbiosis or opportunistic bacteria and Candida. All right, so with these test results in hand, that helps us kind of hone in on a particular type of approach. So for practitioners, it can often be confusing when there's various different types of pathogens and opportunists present as to how do you approach that? Which one do we go after first? How do you address the big picture? So of course we know since there's inflammation involved in certain pro-inflammatory types of microbes really thrive in inflammation, we want to do everything we can to try to reduce that inflammation and the inflammatory inputs, which of course means an anti-inflammatory diet, likely gluten-free, dairy-free diet, possibly lower carb in this case due to the presence of Candida. Basically addressing H. pylori, the opportunistic bacteria and Candida would be important, as well as supporting the beneficial bacteria, supporting the intestinal barrier, and possibly also supporting digestion, maybe more emphasizing stomach acid versus the digestive enzymes, which were normal in this case. Just kind of a quick summary. I won't go through all the details here, but basically this gives you some ideas of how these various types of microbes are often addressed. So for H. pylori, so here over in the left column, we have the various targets, H. pylori, inflammatory dysbiosis, and Candida. It certainly becomes a little bit tricky sometimes to understand how do we address this when they're all present. But basically, we'll just walk through that briefly. But under the approach, it's often common to include something called mastic gum to address H. pylori, along with DGL, which is basically licorice root extract. Then zinc carnosine is also very common in anti-H. pylori protocols, along with S. boulardii, which has been shown to have anti-H. pylori activity, and then immunoglobulins, including colostrum, which has been shown to help bind H. pylori and reduce it. Certain products that contain some of these ingredients, just as examples, would be Gastromen from Designs for Health, and then Pylorosil from Orthomolecular, contains mostly the mastic gum, DGL, etc. Oftentimes you have to provide the Aspulardi and immunoglobulins separately. For inflammatory dysbiosis, which is pretty common, the approach usually involves including a broad spectrum antimicrobial botanical, typically with berberine or some other type of strong antimicrobial as one of the main ingredients, which also include oregano oil, etc. Aspulardi is also very good for just generally counteracting dysbiosis. Probiotics also have been shown in many studies to help balance the microbiome, and once again, immunoglobulins have been shown to do that as well. You can see here on the side some of the common products that contain good antimicrobial broad spectrum formulations. And then with regard to addressing Candida, caprylic acid can be especially helpful in the context of antimicrobial botanicals. And then also Aspulardi can be helpful in this case, once again, along with immunoglobulins. And so if you're addressing all three of these together, the sort of question becomes, how do you approach that? But basically, a broad spectrum antimicrobial can really address all three of these together, along with Aspulardi and immunoglobulins. And then specifically, a protocol that includes H. pylori, once that is aimed towards targeting H. pylori, should include mastic gum, etc. And then for Candida, caprylic acid, making sure that there's that along with the antimicrobial can be quite helpful as well. And of course, supporting beneficial bacteria and general gut support, as we talked about, can be very helpful. So we've mostly talked about these factors: prebiotics, anti-inflammatory support, butyrate and propionate, as well as curcumin, boswellia, so anti-inflammatories that can basically create a better environment to support beneficial bacteria. This is an example on the side of some of the broad spectrum prebiotics that can be used. Gut support, typically there's a lot of different options that we have. Once again, immunoglobulins can be very important. Butyrate, glutamine, zinc carnosine, something called N-acetylglucosamine, etc. And also vitamins A and D can be quite important, especially for those who are not digesting or absorbing their fats quite so well. So just jumping to the key take-home message from this webinar is really focusing on the fact that addressing microbiome imbalances represents a really promising approach to improving the health of individuals with autoimmunity using targeted nutrition and lifestyle approaches. All right, so thanks very much for listening, and this is my contact information if you'd like to learn more.
Thank you so much, Tom. Really a very, very good presentation. I mean, maybe I'm just nerd and geeky, but I'm so grateful for the current research-based focus for this presentation, and all the studies that you had that you're referencing throughout. Your graphics were fantastic, and I could listen to you all day. Your teaching style is just it just held my attention, and wants me coming back for more. So there's my compliments.
That's so nice of you to say.
Yes, sir. All right, let me get to some questions here. I think that when you come down here to the distillate, we're really talking about cellular health and the disruption of the balance of our cellular health to protect us and nourish us and be able to excrete toxins. But when we think about the gut and the lumen, it's like one cell layer. We're talking about a single thickness of a cell. And all the mighty work that has to do. We could just focus on cellular health, right? So yes, let's protect ourselves. And so that brings me and our attention to really this decade is so pronounced in antibacterials, hand sanitations everywhere, antibacterial soaps, this message that we must eradicate bacteria. And I just wonder how much effect that's had on us overall, and wonder what your thoughts are on that.
Absolutely, yeah. So there's actually quite a bit of emphasis on that as being a contributing factor to not only dysbiosis in the long term, but also immune imbalances and autoimmunity in particular, to the point where it's actually considered, or it's actually called the hygiene hypothesis for why we have such an increase in incidence of autoimmune conditions, other inflammatory conditions, and even asthma and allergy, and sort of the more allergic conditions like that. So it is thought that, especially at young ages when the immune system and the microbiome are developing, that having a certain amount of exposure to, quote, the bad guys or the harmful guys, which can, of course, be tricky, may actually be beneficial. And there are studies actually out there, both in animal models and even in humans, epidemiological studies suggesting that at very young ages, exposure to a certain amount of these pro-inflammatory bacteria are really important to basically stimulate the immune system so that it has, sort of the essential long-term tuning. We talked about that fine-tuned balance.
Yes.
That's really, really important. But unfortunately, because a lot of people have not developed with those exposures, and so they're more prone overall to these conditions, once you're an adult and your microbiome is sort of already developed to a certain extent, and especially your immune system, those kind of exposures may not be no longer appropriate in that context. So it'd be more important to avoid the more pro-inflammatory bacteria, or at least try to address those sorts of imbalances.
Sure. No, great answer. And I think that, we need to be exposed to, you refer to these as guys, we need to be exposed to all of the guys. I think it's like, I see so many parents like washing their children constantly, wiping their hands, washing their faces, changing their clothes, and as opposed to go outside and play in the dirt. And the value, it's like you got two opposite approaches here. And how do you develop resistance? How do you develop your immunity? How do you interact with the world when we walk through with such fear of it?
I think you hit the nail on the head, with the key really being, not to completely avoid all these potential bad guys, but to also focus on, kind of use good judgment in terms of situations that may be more risky than others, but also really building up our resistance and focusing on how do we do that.
In many of the things that you shared about beneficial approaches, certainly probiotics came through, and there's an endless supply of new ones on the market, including megaspores. Are you familiar with them, and do you have an opinion about that?
Yes, yeah. So it's a kind of a newer set of products that contain these so-called spore-forming or soil-based bacteria. So they're essentially what are called bacillus species. So they're kind of roughly in the same group, taxonomic group, as lactobacillus and other related bacteria. So they have some similar properties. For example, they all produce lactate for the most part, which can help contribute to a nice low pH in the gut, which can help reduce, basically inhibit pathogens. Bacillus are actually also really good at, and it's also been characterized in research, in terms of the production of antimicrobial factors. So certain bacteria actually produce their own antimicrobials. And so those can be quite helpful in terms of probiotics when you want to help counteract the bad guys. The other thing to note about these bacillus sort of megaspore type products is that research indicates that they tend to be somewhat more stimulatory to the immune system. So they can potentially be especially helpful when patients or clients have a low functioning immune system and they just need to ramp that up a bit. So I think that they're quite helpful. A lot of practitioners really do find that in some of them I've talked to, actually that's become kind of their go-to probiotic for a lot of their patients. I do think probiotics are very individual. So sometimes you do have to do a little bit of that trial and error. So I don't think they're appropriate for everyone. But I think a lot of practitioners have gotten such good results, especially for some reason when they're used in conjunction with immunoglobulins or colostrum. That seems to be, for a lot of practitioners, a really good combination to help correct dysbiosis.
Great, thank you. There's several more questions that I want to just let people in the audience know that are raising your hand. I don't know what your question is, and I can't call on you, so if you can type your question into the question panel, I'll be able to get to that right away. There's also a question here about fecal transplants not being mentioned, and if you use them, if you recommend them, what do you think?
In the US, so they're really only strictly approved for C. diff, really serious C. diff infections, but there's a lot of activity exploring in kind of clinical trials their use in other conditions. And so I think some of the other conditions where they're very close to being approved, and basically where there's data behind that they can really help improve symptoms, if not the actual underlying pathology, would be things like IBS, various inflammatory bowel diseases, So there's some other scenarios where I think we will start to see that application very soon. But basically, it's fairly restricted in terms of types of practitioners that can do it. So as a functional nutritionist, that's not something I'm able to do in my own practice. And I also think there are some concerns that they still need to work out because we don't know everything about the microbiome. So the concern is, basically when you're transferring from one person to another, you may be introducing kind of an additional risk factor. You may be curing one thing and setting the stage for something else down the line. So there's a few of these unknowns, but certainly for serious conditions where there aren't too many other options, and that risk is considered acceptable. I think it's a great option and it holds a lot of promise in the future for when they can really pin it down to making sure that none of the potential pathogens are present, et cetera, and make it really safe.
Yeah, I agree. And you also mentioned bacteriophages, and so I'll remind our listeners that we had a webinar recently on bacteriophages, and so to get more information on that and review it again, you can come back to our archives and get into that, because I think it's another potential piece of this puzzle. And so I'm going to a couple questions concerning diet. Certainly you talked about anti-inflammatory diets, and kind of the classic gold standard right now is the AIP, the autoimmune protocol. Do you agree?
Yeah, I think for a lot of people that can be very appropriate. There are cases, of course, where that doesn't seem to really address their particular issues in a given situation, and that to me kind of leads me to just a little bit of a digression where one of the big promises of basically microbiome testing and just working with the microbiome is we know that so much of the response to diet and therapies has to do with a specific microbiome that's in an individual. So we already know enough to be able to start to work with that. There's a lot more we need to learn. But basically based on the microbiome makeup, we can tell certain things like, are you eating too much protein for what may be ideal for you? Are you able to metabolize these various potentially beneficial polyphenols? Some people can, some people can't. So if we know that ahead of time, we might be able to be more specific about tailoring a diet to that individual's microbiome.
Hence GI maps, right?
Exactly, yeah.
There's I appreciate the test. I'm familiar with GI maps, and I know a number of our students and graduates have begun using it and feel really good about it. But I think that there's a learning curve to that too. And as much as our students learn about testing as they go through their advanced clinical modules at Hawthorn, there's more to learn. So I'd love to have you back if you would be willing and talk more about just the testing.
I would love to. Yeah, that would be great.
It's interesting to me because I expected, I learned a lot in this presentation and I'm grateful for that, but I was surprised that by the microbiome testing itself, but the GI maps, from a clinical perspective, is that's really what we're looking at, as opposed to more of the DNA structure. But maybe both are needed, but it seems like from a clinical perspective, you're really leading us right into our stool testing.
Exactly. Yeah. I mean, I think there's just so much that we can learn from that. It was even a surprise to me when I started working more with the GI map. That basically there's sort of that initial layer of information that you can get out of it, but the more you work with it and the more you learn about the various markers and how they interact, you can really get a pretty complete picture. And oftentimes that's in conjunction, of course, with other test results that can give more pieces of that puzzle. And of course, you always have to take into account, that individual patient or client's situation, their symptoms, et cetera. So you can't draw too many conclusions from just the test itself.
Yeah.
And certainly don't recommend, treating the test. You're treating the patient and the client. But it is an important piece of the puzzle, I think, to really help connect the dots.
Yes, our intake is what's utmost, and the other things, the information that we gather from testing, etc., help guide us as well. So,
Absolutely.
Staying with diet a little bit, you brought up salt, and you mentioned the studies. I haven't read those studies particularly specifically, but for the salt, it was like high salt intake. So, what constitutes high salt, and does that also equate to potassium deficiency?
That's a good question. So I would have to say that's not an area that I'm quite as knowledgeable as I'd like to be. But basically, again, I think it's going to be somewhat individual. There's all kinds of factors in terms of how your body individually regulates salt. For example, I know in my own case I have certain SNPs that make me lose salt pretty quickly. So I generally have to consume more salt to keep my levels up. But I've always been concerned, is that having an impact on especially my probiotic bacteria, because I tend to be deficient in lactobacillus. So that could be a consequence of consuming too much salt in that context, but I actually need to consume more salt for other reasons. So I do think it's a bit individual. But I think if a patient or client is lacking in lactobacillus, and especially if they have an autoimmune condition and there's no other reasons medically or clinically why we can see that they may need to restrict their salt, they're not prone to high blood pressure, et cetera. Maybe a safe context to either reduce their salt if needed or increase their salt, in various contexts. That's you always have to take those trade-offs into account in terms of what aspects of health are you most focused on.
I would agree with you, definitely. But, certainly at Hawthorn and definitely in our macro and micronutrient courses, we're studying about sodium and potassium and the ion pumps, etc. But we're talking about types of salt and how salt is produced and the differences of that. And my understanding certainly is highly commercial processed salt that's been bleached and treated with high heat and chemicals does not any longer act as a solvent in the body, right. and it constricts and it keeps things from flowing in and out of the cells, where a pure form of salt really acts as a solvent, and it helps move the nutrients in the waste out. So I think quality here really needs to be emphasized, and it's often lacking in studies. It's like, yes, they're talking about salt, but they're not talking about the quality of the foods or this kind of salt, and I think it matters so much. And so, a specific question here is like, would sea salt also affect autoimmunity negatively? Well, 95% of our salt is sea salt, so it comes back to how it's processed, I think. But would you distinguish between quality of salt or sea salt of having a different effect on autoimmunity than commercial salts?
I would imagine it does. Now, of course, there's not a specific study to support that because, again, in most studies, they're using very specific purified ingredients. And that is one of the major downsides of any kind of research. For research purposes, they need to keep all those factors very controlled. So they need to know exactly what everything is, what very purified sources, et cetera, which is kind of the antithesis of sort of a natural dietary approach.
Sure.
So sometimes it is a little bit of a stretch to make conclusions from studies. Of course, that's where I think the clinical studies come into play quite a bit. But even then, there's that concern over most clinical studies, which they consider the random clinical trial as sort of the gold standard. But you're always looking at averages and statistics versus an individual. And as clinicians, we're working with an individual. And again, I think quality means an awful lot, and I think it is likely that the salt story is more complicated, and that basically if you're using a good quality salt, for example, sea salt, for example, versus just a purified salt, there may be generally a lower risk that's going to have a negative effect. It's a little hard to state that emphatically without some sort of study to back that up, but that's just sort of my intuitive sense that that's likely to be a better scenario.
Well, from your research perspective and your clinician's perspective, I appreciate your insights and your intuition on that. So go with it.
Thank you.
All right, that's the end of our questions. So I think we're just about ready to conclude this presentation. Do you have any closing thoughts that you want to leave us with, Tom?
I would just say, because we know that gut health is so important and central to health. And if we're talking about autoimmunity, we know there's such an interaction with the immune system, that really thinking about how you can accentuate and enhance your gut-centric approach to therapy, I think is really important. And I think it really is, even though there's a steep learning curve, I think it is well worth considering incorporating more types of microbiome testing or gut testing in your practice if at all possible. My experience has been, and it's just really yields a lot of great insights and actionable information, not only in terms of helping to better understand what's going on with your patients and clients, but also in particular with how do you support them and how do you improve their health. It really, I think, gives a great return once you make that investment in learning how to better understand these types of testing and incorporate it in your practice.
I absolutely agree. Very well said. Thank you so much, Tom, for this.
My pleasure.
I have a few closing comments, and so we'll go through that for a minute. I want to remind everybody that this webinar was recorded and it'll be available up on Hawthorn's website under archived webinars in just a few days. And there's a survey to fill out after the webinar ends, and it always helps us to have your feedback and any comments about this presentation and the work that we do at Hawthorn. So thank you for taking time for that. And just a heads up about our next webinar will be Tuesday, December 4th with Glenn Depke, traditional naturopath, and HU Hawthorn University faculty advisor. He's going to be addressing the topic of ketogenic diets with us. And then a reminder of our next All About Alumni series presentation will be December 5th at noon Pacific time with one of our doctoral grads, Wendy Turner Larson. She's going to be presenting on her postgraduate activities, and it's just, I'm floored by what she's doing, and I'm thrilled to have her on and feature her, and look forward to you learning from her as well. So like I said, that now does conclude today's presentation. So thank you again, Tom.
It was my great pleasure. Thanks so much for inviting me to speak today.
Absolutely. And thank you everybody for sharing in this educational experience with us. I want to wish you all the best of health as we embark on some significant holidays, and I look forward to learning more together at Hawthorn University's webinar and All About Alumni. Take care, everybody.